Hi, welcome to the channel. So, what if I told you that scientists now can edit the genetic code of life itself, bringing back species that vanished thousands of years ago? In October 2024, two wolves cubs named Romulus and Remus were born, but these weren't ordinary wolves. They were genetically engineered recreations of their wolves, which were extinct for 13,000 years. Welcome to the revolutionary world of genetic engineering and de-extinction. This isn't science fiction anymore. It's happening right now in laboratories around the world. Today, we will explore how Colossal Biosciences used cutting edge genetic technology to recreate an extinct predator and understand the incredible science that makes this possible. But first, let's understand what genetic engineering actually is. Now, let's understand the genetic engineering. Think of DNA as a massive library containing of millions of books. Each gene is a specific chapter with detailed instructions. Traditional evolution takes millions of years to rewrite these instructions, but genetic engineering allows scientists to act like editors of this biological library. They can identify specific chapters or genes and rewrite them in a matter of months. The breakthrough technology making this possible is called CRISPR Cas9, often referred to as genetic scissors, discovered by French scientist Emmanuel Carpenter and American scientist Jennifer Doudna, who won 2020 Nobel Prize in Chemistry for this work. CRISPR works with incredible precision. It can locate specific DNA sequences among millions of genes, letters, and cut them in like scissors, then replace them with new sequences. But genetic engineering for the extension involves three main approaches. The first one is backbreeding, where scientists selectively breed animals over many generations to gradually recover lost traits. Second is cloning, which creates identical genetic copies using preserved cells from the genetic animals. The third is direct genetic engineering, the most advanced method that directly edits DNA sequences to recreate extinct characteristics. The Dire Wolf Project, Genetic Engineering in Action. The Dire Wolf De-Extinction Project showcases genetic engineering at its most ambitious. But before we dive into science, let's understand what made direwolf unique. These ancient predators lived across North America until 13,000 years ago. They were 25% larger than modern wolves, with longer heads, wider jaws, and nearly white fur. In a 2021 study revealed that werewolves had separated from other wolf species 5.7 million years ago and never interbred with grey wolves or coyotes. The, the genetic engineering process began with scientists' DNA extraction. Scientists worked with two precious fossils, a 13,000-year-old tooth found in Ohio's Sheridan Cave and a 70,000-year-old skull from Idaho's American Falls Reservoir. Using advanced extraction techniques, they recovered DNA fragments that provided 500 times more genetic data than previously available. However, this still represented only a fraction of what there was genome. Here's where the real genetic engineering magic happened. Scientists used gene sequencing technology to map out the ancient DNA and compare it to grey wolf DNA. The discovery was remarkable. They were 99.5% similar, making genetic modification feasible. Using sophisticated computer analysis, scientists identified exactly which gene controlled the key difference between direwolf and grey wolf. They focused on 13 specific genes that determined body size, head structure, jaw strength, fur color, and, and texture. These genes were like the crucial chapters in the instruction manual that needed rewriting. The CRISPR Cas9 system then went to work like molecular scissors. Scientists programmed the CRISPR technology to locate these 14 genes in gray wolf DNA and make precise edits. This involved 20 individual alterations across the selected genes, each change carefully designed to recreate specific direwolf characteristics. The process didn't stop there. The edited genes were then cultured in laboratories and growing hybrid embryos carrying the genetically engineered direwolf traits. Out of 45 embryo implantation attempts across multiple surrogate dogs, only two embryos successfully developed. These became Romulus and Remus, born through C-section in October 2025 to prevent complication from their potential larger size. The science and its limitations. But here's the fascinating scientific reality. These aren't true direwolf. They are genetically modified grey wolf carrying engineered traits designed to mimic the direwolf characteristics. Out of millions of genes in a complete genome, only 14 were altered, less than 0.000% of total genetic code. This highlights both the incredible power and current limitations of genetic engineering. While we can successfully modify specific traits with remarkable precision, recreating an entire extinct species would require understanding and replicating its complete genetic blueprint, something far beyond our current capabilities. To put this in perspective, humans and chimpanzees share 98.8% of their DNA. If scientists modified 14 chimpanzee genes to create more human like features, would that create an actual human? Absolutely not. Similarly, these genetically engineered wolves represent an impressive scientific achievement, but they are fundamentally modified grey wolves, not resurrected dire wolf. Technical challenges and future implications. Modern genetic engineering faces several significant technical challenges. Ancient DNA samples are almost always incomplete and damaged. Editing millions of gene sequences and simultaneously remains incredibly complex. And perhaps most importantly, we still have limited understanding of how genes interact with each others and with environmental factors to create complex biological systems. Genetic engineering represents one of humanity's most powerful scientific tools. The Direwolf project demonstrates both our remarkable capabilities and our current limitations. As this technology advances, and the real question isn't whether we can engineer new organisms, it's how responsibly we will use this incredible power to shape the biological world around us. If you like this video, please like, share, and subscribe. Thank you.